The high demand for innovation in biotechnology creates new opportunities for advanced products in the life sciences market. At 26 Incorporated, our focus in the life sciences is on manufacturing components and sub-assemblies for analytical tools that interact with light, such as those used to amplify and analyze genetic material in a process called the polymerase chain reaction, or PCR. PCR was invented in 1983 and won a Nobel Prize for American biochemist Kary Mullis. Today, it is a widely used technique in molecular biology for quickly replicating small fragments of DNA or other genetic material into large enough samples for detection, diagnosis, or further analysis, such as genome sequencing. The PCR process is also used in fields as diverse as forensics to solve crimes using DNA evidence, and public health to test people for COVID-19 and other diseases. PCR often involves a precision instrument called a thermocycler. Test tubes containing a mixture of genetic material and specific reagents are loaded into the PCR machine, and the machine cycles the temperature of the samples very precisely and very quickly, over and over. The thermocycling serves to separate or denature double-stranded molecules into single strands, and then rebuild or anneal them into exact copies of the original template using complex enzymatic reactions. The denaturing, annealing, and extension processes caused by the rapid heating and cooling cycles work exponentially such that millions of copies of the original molecule can potentially be created. Thermal engines from 26 that drive this rapid cycling are the state-of-the-art in chiller technology and thermal control, achieving precise and very uniform temperature shifts, as demanded by this technique. Along the way, light-sensitive molecules can attach to the replicated DNA and will fluoresce when excited by a light source such as a laser or LED. The illumination light passes through an optical illumination assembly, often consisting of an array of specialty filters, lenses, mirrors, and beam combiners and splitters that propagate and process the fluorescent signals. Multiple signals at different wavelengths can be combined to illuminate the DNA sample. The wavelengths are then separated and detected by a special imaging device that will transfer the fluorescent signals into a computer for analysis and readout. 26 manufactures the light sources, such as lasers and LED modules, as well as the advanced filters, lenses, mirrors, and other components that make up the signal detection pathway used in PCR. 26 designs the optical and thermo components and sub-assemblies for PCR instruments to suit its customers' specific needs. Manufacturing is also completed in medical-grade clean rooms, qualified to ISO 13485 at 3 different sites around the world to meet the exacting standards of biomedical equipment. Customers value 26 for its vertically integrated production capabilities and its reputation for quality and reliable performance, enabling life sciences researchers and medical personnel to develop life-saving treatments faster and more efficiently using PCR. At 26, our focus on innovation and excellence in the life sciences is part of our company's vision to enable the world to be safer, healthier, closer, and more efficient.